So I'm Deepa Agashe. I'm going to be the host and moderator for today's session. And I'm very, very happy to introduce you to Pleuni Pennings. Pleuni is from the Netherlands originally and did her PhD in Munich, after which she did her postdoctoral research at Harvard and Stanford. And this is where I first met her when she began working on evolution with viruses and evolution with drug resistance. And before that, she has worked a lot on population genetics, various questions in population genetics, trying to understand how populations evolve. And she's now a faculty member at the San Francisco State University, and her lab studies virus evolution. She's primarily a computational biologist, and so she, tries to understand how best to analyze data and make sense of data to understand questions about evolution. So, Pleuni, I'm very excited to have you here, and look forward to your talk.
Thank you, Deepa. It's a pleasure to be here. Thank you all for joining. I'm excited to share some of my work with you. And I'm going to try to share my screen with my slides. Okay, so I hope you can all see my screen.
Yes, we can.
Great. And apologies, just a second. Okay, so I'm going to use my screen, which is on my iPad, and I'm going to sometimes draw on it to try and help you follow what's happening. So my talk today is about drug resistance in viruses, well, specifically HIV, and also the speed of evolution. So how fast does HIV evolve within patients? As I'm sure you're all aware, in 2020, we have learned again that viruses are super important. We are doing this talk on Zoom, and most of us are probably sitting in our homes because there's a virus going around that is dangerous. In the 1980s, HIV was a new virus that we didn't know much about. And HIV became known as a virus that evolves very quickly. And viruses can, but don't always, evolve quickly. Evolution in viruses is relevant specifically for treatment and also prevention. So if a virus becomes resistant to the drugs that we're using, then the treatment doesn't work anymore. And for prevention, we use vaccines, among other things. But if a virus changes and the vaccine doesn't work anymore, then of course the vaccine needs to be changed. And that's why in influenza, you have to get a new vaccine every year. So evolution in viruses is, I think, super relevant. And of course, for something like coronavirus, an important evolutionary step that virus took was to jump from one host into humans. So many reasons to study the evolution of viruses. And my specific research, as I said, is on HIV and drug resistance. So HIV treatment is a success story. In the 1980s, we didn't know anything about this virus. We had no drugs. But nowadays, there's very good drugs, and they're available to many people, though not all people who need it. So this map shows for every country, roughly how many people in the country have access to HIV drugs. And so you can see that in some places in the world, like in Europe and in Southern Africa, it's pretty dark blue, which means that most people have access to HIV drugs. And then, of course, there's also countries with some lighter colors, like India, where about half of the people who would need HIV drugs have access. Now, this is in 2017, so I would hope that now it's 3 years later, the situation could have improved a little bit. But until just a couple of years ago, this looked very different and very few people had access. So this is still a big improvement. And treatment is so important because it saves lives. So here, this is data from the United Nations. You see here over time from the 1990s to 2016. It shows in blue the number of people who died of HIV. And so you can see it peak in 2005 and then it goes down. And then in red, you see the number of deaths that have been prevented by HIV treatment. And so you can see that in any 1 year, there's about 1 million deaths averted due to HIV treatment. ART here means HIV treatment. And so this again shows that HIV treatment is super important and is saving lives all over the world. In the 1980s, there was no treatment. And so this is like a little history based on people who are somewhat famous in the United States. This person is an actor. His name is Rock Hudson. He got HIV in the early 1980s. He was diagnosed in 84. At that time, there were no drugs, nothing they could do. And so he died in 1985. A couple of years later, things started to look a little bit better, but it wasn't good yet. So Freddie Mercury, and I hope you all know who this is. And if you don't, I highly recommend that you look up who he is, because he's an amazing singer and musician and showman. Anyways, he was diagnosed with HIV in 1987. At that time, there were drugs available. And since he was rich and famous, he probably got access to those drugs. But what happened at that time is that the drugs didn't work very well, and the virus became resistant to the drugs very quickly. And so even though there were drugs, they didn't really work. And he died a couple of years later. Here's some data from that time. Here, this is data on about 20 patients in the early 1990s. Here on the x-axis, you see the number of weeks they've been on treatment. So the whole graph is about 3 months. And then here on the y-axis, you see the percent drug resistant. So what that means is that it's the fraction of the virus inside that patient that is resistant to the drug that they were taking. These people were just taking one drug, which is probably what Freddie Mercury was doing too. And so what you can see here is that at the beginning, when they start their treatment at week 0, almost all of these dots are near 0%, which means that the virus was not resistant. None of the virus in these people was resistant to drugs. But then just a couple of weeks later, all of these curves go up real quick. And so after a couple of weeks, almost all of these dots have reached near 100%, which means that all of the virus inside these people's bodies had become resistant to the drug that they were taking. And after 3 months, all of these dots are up here, meaning that all of the virus is resistant. So even though there were drugs, after a short amount of time, the virus became resistant to the drug, and the drug became absolutely useless. And so that's why people like Freddie Mercury died even though they were getting treatment. Things started to improve though in the mid-1990s. This is Magic Johnson. He's a famous basketball player, and he was diagnosed in 1991. And at the time, he thought that he was going to die real quick. So he went home, he retired from basketball, and he went home basically to be with his family and to die. But treatments improved at that time, and he probably got treatment early on and switched treatment a few times. But then in 1995, we got much better treatments that became available in the United States and Europe. Specifically, protease inhibitors were discovered, which is a specific drug that inhibits protease, which is part of the HIV genome. And that allowed us to make triple drug therapy, which is a therapy consisting of 3 different drugs. David Ho was one of the people who who came up with the idea of combining these different drugs. And it was recognized at the time that that was super important, and he was made Time Man of the Year. The result was that in the United States and in Europe, the number of people who died of HIV, which had been going up steadily over time, wait, you can't see that, had been going up over time. Now in 1995, with these new drugs, the number of people that died suddenly went down. And so these new drugs had a huge impact. And for Magic Johnson, it meant that he could survive, and he's still alive today. This effect, we saw it in Europe, in the United States, and Canada, and other rich countries, but in the rest of the world, we had to wait much longer. This is actually data from India. And here you can see that the number of people who died of HIV kept increasing until 2005, and only then it went down. So it took much longer because these good drugs became available to people all over the world. And actually, India played an important role in making these drugs available, not just in India, but also in other parts of the world. One of the last famous Americans who was known to be infected with HIV is Charlie Sheen. He's an actor. No need to look him up. I don't think he's that important. But he was diagnosed probably in 2011, and he got on a very good treatment almost immediately. And then what he said when he announced to the media that he was HIV infected was that his viral load was undetectable. So that's what the newspapers wrote, that he had undetectable HIV. What that means is that the drugs work so well that the virus can almost not replicate inside his body. And so with any regular HIV test, you could actually not detect the virus in his blood. What happens in such a patient is that here is plotted the amount of virus in the blood, which can start very high. This is like 10 million viral particles per milliliter of blood. And then over time, when you're on the right treatment, this number will go down and down and down and down until at some point the virus cannot be detected anymore. This is very good, first of all, because it means that the person who is infected will stay healthy, because the virus cannot do the things that it does to make you sick. Also, it means that you cannot infect other people. So it's also a form of prevention to stop new infections. Another way of prevention is also super useful. HIV drugs can also be used as PrEP, which stands for pre-exposure prophylaxis. This means that people who know that they are at risk of getting infected, they can take this kind of pill once a day to prevent infection. This is important, and there are some cities in the United States, including San Francisco, where I live, that are trying to encourage a lot of people to take this pill. And the result is that the number of infections in the city is going down steadily, and they hope that at some point it will hit 0 new infections in San Francisco. Okay, so here's a brief summary of HIV treatment, of the history of HIV treatment. So in the 1980s, we had no drugs. There was nothing. Then in the 1990s, early 1990s, there were drugs, but they didn't really work. And the reason was that the virus evolved quickly to become resistant to these drugs. 1995, things improved in the United States and Europe with 3 drugs at the same time. And that made it harder for the virus to become resistant, to evolve to become resistant. And we'll talk more about that. In the 2000s, drugs became available that actually were able to prevent almost all evolution of drug resistance. In the 2010s, drugs started to become available in other parts of the world. And also drugs became available to prevent HIV infection. So I think all in all, HIV drug treatment is a real success story, even though it took a couple of decades. So let's switch to the drug resistance in HIV and more the genetics of drug resistance in HIV. So one of the reasons why I can study evolution of drug resistance in HIV, even though I don't have a laboratory, I work only on my computer, I use sequence data, I analyze sequence data. The reason I can do that is because we know a lot about how the sequence of the virus relates to what the virus can do. So we can read the sequence of the virus. And for example, we know that if there is at this specific position, there is an A, that means that the virus may be susceptible to a certain drug. But if there is a T at the same position, we may know that this virus is resistant against a certain drug. Right? So we can translate the sequence data to what we call phenotypic data, which means whether or not, in this case, it means whether or not the virus is resistant to a certain drug or not. So what this means is that if a doctor is trying to decide what kind of drugs to give to a certain patient, they may send some of the virus to a lab to sequence the virus. And then the sequence can be, that's just a bunch of letters, A's and C's and G's and T's. We can paste it into this website. And then the website will basically spit out a list of drugs and whether or not that virus is resistant or susceptible to those drugs. So for example, with the sequence that I put in, you can see that for this drug, the virus is susceptible. For this drug, it's susceptible, etc. But for this drug, the virus has high level resistance. And for this drug also, it has high level resistance. And so a doctor can then look at this list and decide which drugs to use for that patient. It also means that if I have sequence data from the virus of a patient at different time points, I can follow and see which mutations occur in the virus and how the virus becomes resistant over time. Another thing that is not good for patients, but that is convenient for me as a researcher, is that when someone is infected with HIV, they will stay infected forever. There's no cure for HIV. And so if you can follow the patient over time, you can follow the same virus and how it changes over time. And if this person is getting treated with drugs, we know that that may change the evolution of the virus in that person. This is very different from many other viruses, right? If you get infected with influenza, you will be cured. Like you may get sick, but then if you're good, if you survive, then a couple of weeks later, you'll be rid of that influenza strain. Next year, you may get infected again, but it will be a new strain, right? And so if we want to study influenza evolution, we cannot simply follow one person over time. But in HIV, we can. And so if we have data on many patients, we can follow how the virus evolves differently in these different patients. So here are some of my research questions. First of all of my research, all of the things that I'm going to talk about here today is about when treatment is with 3 drugs. Because since 1995, virtually all treatment has been with 3 drugs. So I'm interested in when treatment is with 3 drugs, does evolution of resistance happen one mutation at a time? So does the virus become resistant first to one of the drugs, then to the second, and then to the third, or does it become resistant to all 3 at the same time? I'm interested in knowing which mutation occurs first, and if it's always the same mutation. Does resistance evolution happen mostly through soft or hard sweeps? You may not have heard of soft or hard sweeps, that's fine, I'll explain it later. And then in the end, I'm interested in, can we say anything about the speed of evolution? Is HIV evolution fast or slow? So the first question was, does resistance evolution happen one mutation at a time? And some of the answers we wrote up, Katie Williams and I, she was one of my students in this paper that came out earlier this year. And here is a bunch of data that may look very complicated, so I'll try to walk you through it. So all of this data is from one patient. This patient is called patient 57. They were part of a trial, and they were treated with 3 different drugs. And one of those drugs at the time was a new drug. It's called efavirenz. This is in the late 1990s. Every row, let me see, every row here is information from one viral particle from the blood of that patient. Okay, so here at the first time point, so that's day 0, it's when they start their treatment, we have data from about 10 different viral particles, and they were sequenced. So we determined the genetic sequence of the RNA of the virus. I'm not showing the entire sequence, I'm only showing parts that are relevant for drug resistance in this patient. These numbers are the numbers of the codons that encode the amino acids that are relevant for drug resistance. We're looking here at 2 different genes, the protease gene and the reverse transcriptase gene. They are right next to each other. And for a long time, almost all HIV treatment was either through protease inhibitors or reverse transcriptase inhibitors. And so the resistance that we see happens in these 2 genes, because those are the ones that are being targeted by the drugs. Just a second. I want to take a sip of water. Okay, so here at day 0, almost all of these letters are white, which means that there is no drug resistance. They are like the regular sequence that you'd see in any other HIV virus. But there's a few that are colored, like this one and this one, and those are actually resistance mutations. And I think I see something in the chat. Is that right? Oh, thank you. The mutations that I colored are the ones that cause drug resistance to the drugs. And so at day 0, there's a few viral particles that have resistance, but most do not. And then at day 6, we only have data for 2 viral sequences. But then at day 85, so that's a couple of months after they started treatment, there's this whole block here. All of the viral particles carry either a C or a T at this position, even though they carried an A here. And the C and the T actually, even though they're 2 different letters, they do the same thing. They create resistance to a certain drug. If they occur at this position, in the 103rd codon of reverse transcriptase, they cause resistance to a drug called efavirenz. And so what we see here is that at day 85, the virus in this patient is resistant to efavirenz. Day 113, not much changes, but then at day 169, we see this block here. And almost all viruses now carry this C. And that happened in position in codon 82 of the protease gene. And so this actually causes the virus to become resistant to protease inhibitors. So now at day 169, the virus is resistant to 2 drugs. Here it was resistant to 1 drug. And at the very beginning, actually, it was resistant to 0 drugs. And then finally, at the last time point that we have for this patient, there's this one that comes up. It's in position 184 of the reverse transcriptase. And actually, it makes the virus resistant to another reverse transcriptase inhibitor. So at this point, the virus is resistant to 3 different drugs. So what we see here in this one patient that was followed over time is that the virus became resistant to all 3 drugs, but it happened in a stepwise fashion, first to one drug, then to a second drug, and then to a third drug. And when we looked at data for many different patients, we saw almost always the same pattern. The virus became resistant to drugs, one mutation at a time, one drug at a time. Okay, so if we know that there's one mutation at a time, a next obvious question is, well, what's the order of these mutations? Do we always see the same mutation occur first, or is it a random order? So which mutation occurs first? So here is data from a big group of people, about 320 patients. They were treated with nalfinavir, 3TC, and D4T. This is a protease inhibitor, and these are reverse transcriptase inhibitors. Their virus was sequenced after about 2 years of treatment. And we split these people up into 3 groups. Here are the people whose virus has 0 resistance mutations. Here are the people whose virus has 1 resistance mutation, 2 resistance mutations, and 3 resistance mutations. So what you see is that if you look at all the viruses that have 1 resistance mutation, it's always 3TC resistance and never D4T resistance and never nalfinavir resistance. Then if you look at the people whose virus has 2 resistance mutations, it's always 3TC plus nalfinavir. And I understand that these drug names may not mean anything to you, but what is important here is that you see that if there's 1 mutation, it's always this one specific one. Whereas if it's 2, it's always this combination of 2. And so what this data suggests is that the virus always takes the same evolutionary path. First, there's no resistance. Then it gets 3TC resistance. It adds nalfinavir resistance. And finally, it adds D4T resistance. So for some reason, the virus accumulates resistance mutations in a predictable order. But that order is actually not always the same if you look at different treatments. So here I have similar data, but these are people who were treated with efavirenz plus 3TC plus ACT. So 3TC was always also present in the previous slide, but efavirenz is actually also an RT inhibitor. So what you see here is that here are the people who have no resistance, or their virus has no resistance. And then here are the people whose virus has 1 resistance mutation. And in almost all cases, it's efavirenz resistance. And rarely it's 3TC resistance. But then when you look at the people whose virus has 2 resistance mutations, you see that it's almost always a combination of 3TC and efavirenz. And then finally, here are a few people that have resistance to all 3 drugs. So let me go back to that previous slide. So what you see here is that for this set of drugs, the first mutation to occur in the virus is almost always 3TC resistance. Whereas with this set of drugs, the first resistance mutation to occur is almost never 3TC resistance. And so it turns out that the order of mutations is predictable for a given set of drugs. But if you change which drugs people are on, it may change the order in which the mutations occur. So which mutation occurs first? I think we can say, well, it depends on the combination of drugs that you're using. So next, my question was, does it happen mostly through soft or hard sweeps? So when I say sweeps, I mean selective sweeps. And so I looked up what Wikipedia says about selective sweeps. And I'm going to read here this part in blue. So a selective sweep can occur when a rare or previously non-existing allele, an allele here is, I had referred to this as a mutation. And mutation is thought to it creates a new allele. So you could read here, when a rare or previously non-existing mutation that increases the fitness of the carrier. So the carrier here is the virus. And the fitness here means whether or not it can replicate or how fast it can replicate. And so when someone is treated with drugs, a mutation that creates resistance to the drug makes that the virus can replicate faster. So it increases the replication fitness of the virus relative to other members of the population, so relative to the rest of the virus in your body. And then what happens is that mutation increases rapidly in frequency due to natural selection. And so what you can see is that, and we saw it in some of the previous slides, is that a mutation occurs and quickly the entire viral population will be dominated by that mutation. So that's what we call a selective sweep. And so here is an example from another patient from that same data set from the late 1990s. This is patient 86. What you see here is at day 0, there's no resistance. This G is in blue, but it doesn't cause any resistance. These other dots here are mutations, but they are mutations that do not cause drug resistance. These are mutations that probably don't really matter for the virus, but they just occur naturally. At day 84, suddenly all of the virus carries this T mutation. This is again that mutation at position 103 that makes the virus resistant to efavirenz. And so the virus is now resistant to that drug. But then what you also see is that here there appears to be sort of some variation. Some of the viral particles carry a mutation, whereas the others do not. But at this point, almost all of the viruses carry the same mutations, even though we think that these mutations are not necessarily important. And the reason is that whichever viral particle happened to get that T first, whichever mutations just happened to be present on that viral particle, they increased in frequency together with that T. So they were lucky, not that they care, but you can consider them lucky to be present in the same virus where that resistance mutation occurred. And then as the resistance mutation is able to make the virus replicate, these other mutations replicate along with it. And so we call these mutations hitchhiker, these ones, the ones that probably don't matter, but they increase in frequency together with this important mutation. We call them hitchhikers. And this can be called a driver mutation. And so this is a hard sweep because here all of the variation is lost in the sense that either mutations go to 100% frequency, they are seen in all viruses, or they are not seen in any viruses. This is an example of what we call a soft selective sweep. Here, again, at day 0, things look very similar. There's some variation here. There's no resistance here. But then at day 28, so this is very quick, we see a T here and a C here. Now, functionally, the C and the T here are doing exactly the same thing. They make the virus resistant to efavirenz. But because we see 2 different mutations, we know that these mutations must have occurred in 2 different viral particles in the body of that patient. And maybe even more of them happened. But at least twice a resistance mutation must have occurred in this patient. And so what we see now is that some of the viral particles carry the T and some carry the C. And the result is that here, a little bit more variation is retained because some mutations were able to hitchhike together with the T and some were able to hitchhike with the C, and they're not necessarily the same ones. So this is what we call a soft selective sweep, when there is at least two different origins of the resistance mutation. And here I'm going to try and show a short video. It's just 3 minutes, but I have to stop sharing here and share a different screen. And this video is made by my colleague Alison Feeder, who I did this work with together. Let's hope that this works.
When the first HIV drug, AZT, was FDA approved in 1987, drug resistance to AZT emerged in practically all patients during the first year of treatment. Nowadays, HIV patients take combinations of multiple drugs, and resistance evolves very rarely. What makes these combinations so good? When resistance does evolve despite the fact that combinations are good, how does it happen? When an HIV patient starts treatment, they generally have a genetically variable population of viruses. If the emergence of drug resistance is rare, a drug resistance mutation occurs in just one of these viruses. As the drug resistance mutation spreads through the population, so does the genetic background on which it arose. The result is that the population with drug resistance is much less variable than the population without drug resistance. This is known as a hard selective sweep because the variation is swept away almost completely. Now, imagine mutations are more common, and we can see resistance mutations happen on multiple genetic backgrounds. If they all spread, then the resistant population may be nearly as diverse as the original population. This is called a soft selective sweep, because while some variation is removed, much remains. Because bad treatments are associated with common evolution of resistance, mutations must happen often, and we thus expect soft sweeps in those cases. For good treatments, resistance evolves rarely, so we might hypothesize that mutations that lead to resistance to complex combinations of drugs are much less likely to occur, and thus, if they do appear, we expect them to lead to hard sweeps. To test this hypothesis, we took data from 6,717 patients from the Stanford HIV Drug Resistance Database. These patients were treated between 1989 and 2013, and we roughly separated the patients into those treated with better combinations of drugs and those treated with worse combinations. We then separated those groups into those whose virus had no drug resistance mutations and those whose virus did have resistance mutations. We found that among the patients on bad treatments, there was no difference in genetic diversity between viral populations with and without drug resistance, as we expect if soft sweeps are occurring. But for the patients on good treatments, we found that the viral populations with drug resistance had much less genetic diversity, as expected if hard sweeps occur. This represents the main finding of our paper. Drug resistance seems to emerge via soft sweeps, implying that resistance mutations are common, in patients given bad treatments, and via hard sweeps in patients given good treatments, implying that resistance mutations are rare. Why do resistance mutations seem to occur so rarely for modern drug combinations? One possibility is that the virus becomes resistant to all drugs present in the cocktail only when it acquires multiple mutations at once, and getting all mutations at once is unlikely. Alternatively, it may be simply because the virus population becomes small if the treatment works well, and in a small population, fewer mutations occur. Finally, you may wonder if this is going to help HIV patients. Well, no, and the reason is that HIV treatments are already extremely good at preventing the evolution of resistance. However, drug resistance is a huge problem in many other diseases, including malaria, tuberculosis, and cancer. We hope that lessons from HIV will help us halt evolution of resistance in those cases as well. Have a look at our paper, now out in eLife.
I'm going to go back to my slides.
Pleuni, there's a question. This might be a good time
yeah?
to quickly ask it. So one of the attendees is asking, is the resistance in 3TC localized at the same position across the different drug regimens?
Yes, it's almost always the same mutation at position 184.
And is it known from sort of random mutagenesis or other experiments whether that particular position is the best position for the virus to acquire maximum resistance, or is that not the case? There are other positions which are also known to be good resistance, offer good resistance.
Yeah, that's a good question. No, I think that mutation is probably the best for the virus to become resistant to 3TC. We do see alternatives at that same position. So sometimes if you have enough samples, you see that first it's a methionine at that amino acid, and first it switches to some other amino acid, and then it switches to the better amino acid. I don't remember which amino acids these are. But so we sometimes see sort of an intermediate resistance amino acid, and then because it's more accessible, mutational, because mutations are more common. And then it gets the better one. But I don't think there's any other known mutations in the genome that make sort of more resistance to 3TC.
Okay, cool. Thank you.
Okay. So the video, what it showed is that when treatments are not very good, and so this is mostly like from the late 1990s and the early 2000s, when people are treated with 3 drugs, but they're not as good as they are now, we see mostly soft selective sweeps. And in later years, when treatments are getting better, we see more hard selective sweeps. And so this is related to the next question. My last question is evolution fast or slow? And so here are some data from the late 1990s for about 12 years. And just a second. This is split in 3 different drug classes, but you see sort of the same pattern in all the classes. What is shown here is the rate at which drug resistance evolves. So this is the per month incidence of drug resistance, meaning it's a probability for each month that resistance evolves in a patient, right? So they say this first point is 0.1. So that means about 10% chance each month that your virus becomes resistant to that drug. And so if it's about 10% a month, after a year, it's almost guaranteed that you have drug resistance, right? So this means that evolution is pretty fast in almost all patients. You'll have drug resistance after a year of treatment. But then over time, that rate goes down. Every year, things get a little bit better. And at this point is about 0.002. So it means that less than 1% chance per month of getting drug resistance, right? And so for these other 2 colors, the rate starts a little bit lower, but also every year it improves. And here the rates have become really low, right? So what this shows is that over time, if you follow patients, people who were treated in 1996 had a very high risk of getting drug resistance. But if you compare with patients who were treated in 2008, they had a much lower risk of getting drug resistance. And actually that improvement has continued. And nowadays the risk of HIV becoming resistant to treatment has become really quite low. And so what this shows is that evolution initially was quite fast, and then over time it became slower. And so this goes together with initially we see soft sweeps when evolution is fast, and we see hard sweeps when evolution is slow. So I like that because I can connect sort of what we know about population genetics and what happens within a population of virus within a patient with what we see in the larger, in what it means for treatment in actual people. So why are things becoming slower, or why are sweeps becoming harder? The reason is mostly because the amount of virus within the patient is getting lower over time. So what we're showing here, this is data from a 2014 paper. This is years, 1995 until 2010. And what you see here is the percentage of participants with undetectable viral load. I hope you remember the undetectable viral load means that the amount of virus is so low that you cannot detect it with a regular test. Okay, and so in 1995, in clinical trials, about 43% of the people have undetectable viral load. Whereas in 2010, so that's 15 years later, almost 80% of the people have undetectable viral load. So what you see is that on average, in all these people, the amount of virus in their body goes down. And if you have less virus, you'll have less mutations. And if there's less mutations, evolution will be slower. And if evolution happens, it's more likely to happen through a hard selective sweep. So here is a little summary. So my question was first, does evolution happen one mutation at a time? Yes, it does. The answer there was very clear. Which mutation occurs first? Well, it depends on the treatment. Does it happen through soft sweeps or hard sweeps? Well, mostly soft sweeps in the 1990s, but hard sweeps now. And then is evolution fast or slow? Well, of course, it depends a little bit on what you call fast or slow, but definitely it was quite fast early on. Definitely when treatment was still with one drug, but even when treatment was with 3 drugs, evolution was quite fast in most patients with a very high probability of resistance evolution within the first year of treatment. Nowadays, it's quite slow, and most people can stay on the same treatment for many, many years without having to switch. So that connects to, these famous Americans. No, sorry, Freddie Mercury obviously is not an American. Fairly famous people. Freddie Mercury probably died because his virus evolved quickly and became resistant to the drugs that he was taking. And so we go from fast evolution to nowadays there's slow or no evolution. And that's why Charlie Sheen can have undetectable viral load and can be pretty sure that he's able to live a long and healthy life. So that's the end of my story. Thank you very much for listening. The story I shared were a collaboration with Alison Feeder, who's now at UW, University of Washington, that is. Kristen Harper, who's a freelance scientist. Katie Williams, who was one of my master's students. These are other people in my lab and funding agencies. And thank you for listening, and I'm happy to take questions.
Thank you, Pleuni. We have one question from Neha, who has raised her hand. Neha, I'm going to allow you to talk. Please ask your question. Neha?
Oh, they're muted still.
Yeah, no, I allowed them to be able to speak, so I'm not sure what's going on. Okay, anyway, maybe Neha, you could type your question in the Q&A box, and meanwhile, we'll take other questions. So one question is, why does HIV virus undergo slow evolution now? Could you explain that a little bit once again, perhaps?
Yeah, so we think that it undergoes slow evolution now because there's simply less virus in someone's body, and evolution needs mutations. And if there is less virus, if there are fewer viral particles that are replicating, then fewer mutations will occur. And with fewer mutations, basically the virus has to wait longer to get to have a chance to get the right mutation. And so, there's a big difference between having, say, 10,000 viral particles per milliliter of blood versus 10 viral particles per milliliter of blood. There's just much less supply of mutations. And so the chances of getting the right mutation are low for the virus.
Do you know whether a similar pattern is observed for any other pathogen as well, other viruses or other pathogens, where at the beginning, when the treatments are not as effective in reducing the population size, they evolve much faster in terms of drug resistance. And then as we get better treatment on the market, then, their evolution slows down, as you explained for HIV.
Yeah, that's a good question. I think we may have a similar pattern in hepatitis C, but sort of the development of drugs for hepatitis C was very fast, and so you don't see it sort of occur over many years. And another case where I know something similar is like for some cancer treatments, some leukemia treatments, where like initially you would get the cancer would become resistant to drugs, and nowadays people can stay in remission for much longer because drug resistance evolution of the cancer has become less likely.
Okay, so we have Neha's typed question. So she's asking, how do we approach treatment when soft sweeps occur? Because so many combinations of drugs will still lead to different mutations because of variation sustaining in the population. So do we understand how best to treat soft selective sweeps?
Yeah, that's a good question. So I think what happened at the time in, say, the late 1990s and the early 2000s is that people were actually working not necessarily to treat soft selective sweeps because nobody knew that that was happening at the time, but rather they were trying to make sure that, say, the drug levels in the body stayed high, or that the side effects of the drugs were minimized. And these two things together meant that the treatments, people were better at sticking with their treatments because of fewer side effects, and maybe they had to take their drugs less often because the drug levels would stay high in the blood longer. And so together, these effects meant that there was more drug pressure on the virus, and so you get less virus, and that sort of automatically solved it, even though I don't think anyone was trying to solve this particular problem, but they were just trying to keep the patients healthy, and it worked by stopping evolution.
And do we know about the spread of viruses? This is, I'm paraphrasing a question from someone watching on YouTube. Is the spread of viruses in cattle or wildlife similar? Does it follow similar patterns as the virus evolves and sort of infects more and more individuals?
Wait, I'm not sure if I heard the beginning of the question.
So sorry. Yeah. So the spread of viruses in animal or sort of wildlife populations, does it follow pretty much similar dynamics as it follows for human populations, for instance?
Oh, yeah. I think in general, like epidemics happen in wildlife as well. Of course, you won't get drug resistance if there are no drugs. So this particular type of evolution will not happen there, and will not happen in most viruses because we don't have drugs for most viruses. But other than that, I think viruses do very similar things, whether it's in humans or in animals. And of course, epidemics are most dangerous when there's a lot of similar individuals close to each other. So farm animals are more at risk usually than wildlife animals.
Cool. There's another reader question from Dr. Sridhari. Why do we not have a vaccine for HIV yet? Why is it so difficult? Yeah.
That's a good question. I wish I understood that, but I don't. Yeah, I feel like I should probably do some more reading on this. I'm not sure why we're able to make a vaccine, say for coronavirus, within a year. We're not able to make an HIV vaccine. It's not because of lack of effort. Many labs have worked on HIV vaccines. Many have been tried, and some of them work a little bit. But I'm honestly not sure why it's so much harder for HIV than for other viruses.
Okay. There's a question from Aruna. Is there any fourth mutation to resistance that is observed in these three drug combinations that you described in your talk, in the first part of your talk? Or are there only three mutations that I've ever seen?
Yeah, no, there are actually more mutations. We just sometimes lump them together because if there's like similar mutations, there are several mutations that have a similar function. They all make the virus resistant to the same drug, for example. We may just lump them together for making the analysis simpler, but there's actually a whole list of mutations. So for each drug, we know of several mutations that cause resistance to that drug. I think 3TC is sort of exceptional in that we almost always see the same mutation.
I guess I have a related question. Is it possible to predict which class of mutation is going to occur first, like the protease versus the reverse transcriptase, depending on what the drug targets? Is it any drug that targets the reverse transcriptase will typically, that's the first one to show up, or not always? Or
yeah, that's a good question. I think it has to do with penetration of the drug in different parts of the human body. So I think that whichever drug has the best penetration, for example, in the brain, that drug will be most vulnerable to drug resistance. So for example, if 3TC can enter the brain, but the protease inhibitor that it's being co-administered with cannot enter the brain, then there's a little part of your body, well, big part, your brain, where the virus only encounters 3TC because the other drug is not there. And so then the virus has a sort of a simpler task. It can become resistant to that one drug that is there by itself first. And then you have a 3TC resistant viral population in your brain, and it can migrate to other parts of your body and become resistant to the other drugs. It's super hard to measure where drugs go in the body. And different people who measure it don't agree with which drugs actually go where. But so I cannot prove this, but I believe that it has to do with how well drugs penetrate into different parts of your body.
Mm-hmm. And I guess a related question is about, maybe this will be our last question. How do we know or understand why protease inhibitors are not, resistance to protease inhibitors does not come up first? The first mutation is never in the protease. Do we know why?
Yeah, so I think it may be because the protease inhibitors do not have don't have the best penetration in different parts of the body. And it may also be because these mutations may come with a cost, possibly.
Mm-hmm.
But so, yeah, we don't know exactly. These are guesses, and these are things that are hard to answer.
Okay. Yeah, I think we should stop here because we are at 5 p.m. and yeah, we should close the session. So Damini, would you please take over?
Yes. Yeah, with this, I would like to thank Dr. Pleuni Pennings for a very interesting talk on the HIV evolution. And despite all the challenges imposed by the virus on mutations in response to drugs, I think with multiple drugs, we are able to combat. And the success story was very fascinating. With all the science we've done, we were able to appreciate it better. And I would like to thank my co-host, Dr. Deepa, for selecting such an interesting topic and a speaker to present it in a simple way that everyone could understand. And I would like to thank the communications team for supporting this webinar for some time now, and also NCBS for the same. And finally, the audience for coming every Sunday over on the Zoom and making this webinar even more interesting with your very intelligent questions. And the conversation is very beautiful to watch. I hear from both the speaker and the co-host. And with this, I would like to once again thank the speaker and the co-host and the audience. We will see you next week, next Sunday. Until then, please take care.
Thank you, Pleuni. See you.
Thank
Thank
you.
you so much. And I'm closing the call. Have a great evening. Bye-bye.
Bye.
Bye.